see if where these R8A located, and it indeed located in the third segment of the antenna. And we also try the specific driver and make sure this is, if we block the specific neurons, this is down regulation of the receptor, this is down inhibition of the specific neuron, and we indeed found that these modulation can inhibit their normal hypotaxis. So next thing is, what's the R8A function in the brain? So we repeat the functional response data in the R60A and R40A and found indeed the same functional response, which the R40A is dry sensor and R60A is wet sensor. So we further found R8A is a wet sensor as well. So there are two wet sensors in the brain. Why there has to be two? So obviously we have to test what's the preference that if we activate R8A can induce. So this time, it's a wet sensor and we activate it, and then this fly shows avoidance behavior. So this is the first lucky part. So remember that when we activate IR68, which is another wet sensor, the fly will drive to the attraction site. And now is the avoidance side. So that means even though they are both sensing the wet stimuli, they have different downstream area in the brain that's sending different signals. And more interesting is if we block, if we make the fly thirsty, now the R8A wet sensing signal is gone. Completely gone, and even look like this, even darker than before. Okay, so this is just to show you the raw image that which area of the brain that of the R8A neuron be activated after wet. So next question is, who is inhibiting R8A in thirsty flies? So we turn into another candidate, which is a glucokinin. This is fly's internal sensor of thirsty. There are only 4 glucokinin positive neurons in the brain, and these neurons their activity will elevate it when the osmolarity changes in the fly body. Okay, and this has been proved in Professor Lin Shuwei's lab back in 2019. So we think glucokinin might be the key, and indeed it is. So we downregulate R8A, we downregulate glucokinin, these R8A neurons, and we kind of rescuing their thirsty behavior. So now the fly doesn't look like they're thirsty, even though they've been, water deprived for like 14 hours or something. And if we look at into the brain, and we do see their brain activity change back to the normal fly even when they're thirsty as well. And we also did some molecular tests. We inject glucokinin peptide directly, and we see similar results. And finally, the most interesting experiment is this one. So in the in the thirsty fly, because we know the R8A should be inhibited now, right? So in these two control fly. But if we artificially activate them, we will see that avoidance strengths that pushing the fly back to the low humidity area. So all these experiments together prove something, that in the fly brain there are 3 different humidity sensing neurons. One is dry sensing IR48, 2 are wet sensing. But IR48 and 60 are responsible for dry fly to wet area, and the R8A is driving the fly to the dry side. Okay, so this is a really cool and fun study, and we eventually published this year. And so the only take-home message here is, as Professor Luo said, never give up. So you always can find something really interesting out of your data, and just, when you look closely, and even somebody else just, publish some of the most important part that you think in your work, there's still a lot to discover, in the science field. Okay, so this is the first part. I hope everybody kind of understand. If you don't, you're most welcome to discuss with me after the talk. And the second part of my talk is about imaging.